So, in our plant science conversation series, the garden's leadership engages in casual discussions with fascinating plant science researchers. In the upcoming programs in this series, we are excited to showcase agricultural science, which connects to our current exhibit, Cultivate Growing Food in a Changing World. Today, Dr. Gehan will be in conversation with our executive director, Dr. Susan Pell, to discuss her fascinating research. Dr. Malia Gehan is principal investigator at the Donald Danforth Plant Science Center in St. Louis, Missouri. Her research focuses on how crops respond to temperature stress. By understanding the natural variation for improved resistance to temperature stress in plants, her lab can help improve crop productivity and better understand how plants perceive temperature. Botanist, educator, and executive director of the U.S. Botanic Garden, Dr. Susan Pell has traveled the world collecting plants for scientific study. Using her expertise, Susan shows people the awesomeness of plants every day. Welcome, Dr. Gehan and Dr. Pell.
Thanks so much, Emily. Really appreciate the introduction and super excited to be here today with Malia. And just really kind of want to kick it off by asking you if you can give us a little bit of an introduction about the Donald Danforth Plant Science Center and the research that you do there.
Sure. So the Danforth Center is the world's largest independent plant research institute. It was founded in 1998, and we are a very international group of scientists. So there are about 400 scientists now at the center, comprised of, there's about 24 labs that all have various research focuses, but we're kind of united under the mission of improving the human condition through plant science. So like the introduction said, our lab is particularly interested in temperature stress as it relates to climate change. So the world's getting hotter. How can plants survive those conditions? And so like we are interested in improving plants' resistance to temperature stress, and the way we do that is to look at how there is variation in how plants respond to that different temperature stress.
Fantastic, thanks so much. And I'm very interested in, you are sort of a traditional plant scientist, but you apply your research within agriculture. And I wonder if you can talk a little bit about sort of the applications of your work in agriculture.
Sure. So what we do is still very basic science. So we're stuck in the lab most days, or we're growing plants in these boxes that can simulate various environments. But the purpose there is to really try and understand what's going on at the molecular level, so that we can then translate that to improving plants and agriculture downstream. So one of the big challenges in agriculture right now is being able to measure how plants respond to stresses. So a pretty fundamental thing, how do plants respond? And so the way we measure plants' responses to stress is to take pictures of them. And that seems like a very simple sort of concept. People have been taking pictures of plants for a very, very long time, or they've been drawing plants. But what's new is that we're trying to take these pictures in a very automated way and then analyze them in a very automated way, so that we can extract information from these images that we previously couldn't.
And can you talk a little bit about that process and how it's
Sure.
Awesome.
So so like I said, everybody's been taking pictures of plants for a long time, or drawing them. And so what's new now, again, is we're automating this process. So we're having robots take pictures, or we're doing time-lapse photography of these plants and watching them grow through time. So we in our lab use a lot of what are called Raspberry Pi computers. So they're actually built for education purposes. They're $35 computers, and you can buy $25 cameras that go along with them. And what's nice about them is because they're very, very inexpensive, we can buy a lot of them. And that means that we can take pictures of lots and lots of plants. And so that's what we do. We take pictures of plants over time and see how they respond to these stresses. And then because we're taking what ends up being hundreds of thousands or even millions of images, we need a very fast way of analyzing that, because we don't want to take student time or graduate student time or postdoc time and having them measure those things by hand. So we need the computers to be smart enough to do that. And so we build an open source software that we call PlantCV to actually extract information from those images.
And I mean, the software that you've developed is really fascinating. I mean, there's so many wonderful applications. I wonder if you could talk a little bit more about it, and then maybe also some of the applications that have been unexpected that other people have used it for.
Sure. So for plant science, the traits that we're trying to extract are ones that you might expect a plant breeder to be interested in. So how big is the plant? How tall is the plant? How early does that plant flower? And so we build these, what are called computer vision, because we want the computer to see these traits, tools to extract that kind of information. How yellow does the plant get under stress, as another trait that we're interested in. So we built these tools, we build them openly. And what that means is that as soon as we build a tool, it's available to the research community, but also to anybody doing research. And so it's often difficult to track who is using your software when you just put it out onto the internet. What we do is we monitor the publications that actually use our software and that cite it. And so we've had some very interesting applications that have been non-plant science applications, even though our software is called PlantCV. And so somebody used PlantCV to measure cracks in sidewalks. They were material scientists, and they were interested to see how the cracks in the sidewalks were looking. And another person wanted to use our software to look at veins in eyes. We had never spoken to these people, but they were using our software for that application.
Yeah, that's awesome. I imagine, like, all sorts of things, river patterns and all sorts of things might be able to be used. You might be able to apply your software to those things, or you, not necessarily you, but other scientists could apply it to lots of other things. So that's really cool.
Exactly.
Yeah. You mentioned temperature stress, and I wonder if you could speak a little bit kind of broadly about plant stress and the different kinds of things that do stress plants.
So plants are amazing because obviously they're having to deal with the outside world. They can't come inside and put on a coat or take off a coat if they're getting too hot. And so they're dealing with a bunch of different environments. There are temperature changes throughout the day. So even the day temperature versus the night temperature, there's often large swings depending on if it's cloudy or sunny. And then there's seasonal changes as well. And plants need to deal with all of those. And if you look outside, there are probably plants growing out in cracks of sidewalks. So they're having to deal with drought or nutrient stress because they're not getting the nutrients they exactly need. So plants are amazing, and they've developed all these different mechanisms to try and survive those various environments. And so our lab, we study plants that are useful for agriculture. So meaning that they are related to plants that we eat, or that are useful for bioenergy, or fibers. So some people at the Danforth Center work on cotton, for example. But we also like to study plants that are really good at growing in these harsh environments. So one of our favorite plants right now in the lab is a plant called quinoa. And you may have eaten it for lunch at some point. But what's amazing about quinoa is that it's adapted to grow in fairly harsh conditions. So you can water it with what is equivalent to about half salt water, and it will thrive. And it also grows at really high elevation. So it can be very cold, but very high light intensity. So very sunny. And so we are interested in this crop plant because it's able to grow in these very harsh conditions, even if the laboratory tools aren't, yet available for them.
And so what do you think might be some of the outcomes of that work, or the applications of that work, especially as we think about the changing climate that we're all experiencing and will continue to experience?
Yeah, so for quinoa, one of its its major limitations for expanded growth in the US is that it's not very heat tolerant, but it's very cold tolerant. And so, we're interested in finding varieties of quinoa that are more heat tolerant. And so say finding genes that make it more heat tolerant. And since it is very salt tolerant, we can maybe find genes that help it be salt tolerant and move those to plants that may be less salt tolerant, or mechanism. So quinoa, for example, has these salt bladders on the exterior of its leaves. So it actually excretes the salt that it takes up, which helps it survive in very salty soils. So can we actually make other plants more salt tolerant using some of these same mechanisms?
That's fascinating. And that research, of course, would lead to being able to grow plants in areas that we can't grow them today, but also looking at how some of the areas that we do grow plants in today might be changing in the future. They might be inundated with salt water, at least, at some parts during the day, et cetera. So really expanding kind of what's land that's available for agriculture, and continuing to make land that's currently available even if the circumstances change.
Right. So that's the big, very big challenging part, is that not only do we need to use marginal land, but we need to make it very, very productive. And the land that we are using that is very productive, we need to keep it productive, even if, even if it's very difficult year to year.
Absolutely. Can you talk about specifically some of the plants that you work on?
Sure. So quinoa is, again, one of our favorite examples. We have projects on sorghum. And we also have projects sorghum, because it's an interesting grain crop for sub-Saharan Africa. Also because it's an interesting bioenergy crop, so there are bioenergy versions of sorghum. We also have projects on corn, because it's obviously a major crop for the US. And that particular project is we're looking at not only heat tolerance, but heat in combination with other stresses. So often when you have heat stress, you might also encounter a drought stress because it's hot and it's dry. And when we're doing basic science like we do in our lab or many labs across the US, it's challenging to study stresses in combination, because number one, it's challenging to study individual stresses. So when you add those things together, it becomes even more challenging. But it's important because you can get a different result under a combination of stresses versus just looking at the individual stresses. So we know that is true, is that when you combine stresses, it's not just the same as looking at the individuals.
Absolutely. And I wonder about the sorghum work that you're doing. I know that there's quite a bit of work being, certainly in this country, but I think globally too, on looking at perennial sorghum. And I'm wondering if there's any connections that you've made with that research.
Yeah, so not perennial sorghum in particular, but we have a project in the lab in collaboration with a lot of other labs, namely Alison Miller's group, looking at various perennial crops. If we're interested in helping climate change and and capturing carbon, then perennial crops are really interesting because they have these very deep root systems that can keep carbon in the ground. And so perennial crops are really interesting for various reasons. They're in relation to other crops, they're understudied because it is more challenging to look at crops year to year to year to year. As opposed to more annual crops that have the seasonality and that you can grow each year. So that's a project that we're really excited about. It just finished its first year, and it also has a large workforce development piece that we're very excited about, because not only do we want for there to be more research on perennial crops, but we want there to be the people that are able to study perennial crops and plant science in general in the future.
Absolutely. And I wonder about the sort of differences for the plant and experiencing stresses, if they're perennial versus annual, and certainly with seasonality. But I'm wondering if there's any other impacts or differences between perennial plants versus annual plants and stress.
Yeah, that's something we're really excited about looking at, and that's actually why our team is a big part of it. Is that we are obviously a big part of the phenotyping piece in collaboration with labs from St. Louis University and other labs at the Danforth Center to really try and look year to year with non-biased phenotyping methods that can be used year to year. So traditional phenotyping methods, somebody going out in the field and measuring height or making some notes about how those plants look. That's really important work. But we're hoping to try and make that a little bit more standardized so that we can see these changes for year to year that are so important for perennial crops.
Excellent. I want to shift a little bit and think about agricultural practitioners. And I know farmers think about plant stress all the time. They're constantly trying to mitigate for with irrigation, with nutrients and all kinds of things. And I wonder kind of if you could speak a little bit to the connections between your research and how a practitioner might be out in the field looking at stress and responding to stress and mitigating stress.
Yeah, so one of the projects that we are really excited about involves something called hyperspectral imaging. So that's if you have a cell phone camera that has 3 channels, a red channel, a green channel, and a blue channel, so 3. Hyperspectral imaging is special because instead of 3 channels, it has 1,000 channels. And that means that we can get a lot more information about what we see in those images. So instead of just seeing that the plant is green, we can tell that, oh, that plant actually has more chlorophyll in this region versus this different type of chlorophyll. So we can actually tell some biochemical properties about that plant. So we're really excited because a lot of remote sensing research has been done to actually image over fields, and so that people can develop ways of picking out, oh, this is an area of that field that is drought stressed or has nutrient stress. And so part of our research is to develop these new algorithms that are more specific. So is it, what type of nutrient stress are they seeing? Not just that it has nutrient stress, but is it nitrogen stress or is it phosphorus stress? And so part of our research really is to connect the biochemical to this image data that we're capturing.
Oh, that's awesome. Really, really cool. We've got a question in the chat that I think is kind of a little bit beyond scope, but it touches on some things that you just mentioned, which are sort of soil conditions and how they impact plant stress and potentially sort of how those conditions might be indicators of other stressors, other stress within the soil, such as it might be impacting, the bacterial, the sort of microfauna within the soil as well.
Yeah, what's challenging about all of this is everything is interconnected.
Right.
So you pull on one string and then you're pulling on the other. So absolutely, soil and soil health are major contributors to what we see in the plant. So the plant can be sort of the canary in the coal mine, kind of often to what's going on in the soil. So if you have unhealthy soil, you're going to see that in the plants that you grow. So they're totally interconnected, and microbiome is absolutely a big part of that. And so again, this perennial project that we're excited about has a big soil health component, and there are soil scientists on this project. For basic research like we're doing, again, that makes our research very challenging because you can have plant phenotypes in one soil and you can have very different plant phenotypes with the same plants in a different soil. And those can change again over time. So the relationship between time and stress is also interconnected with this relationship between soil and plant. So that's why we're employed, basically, is that we need to look at all these different facets, and it's sort of never ending. And that's why plant science is really interesting, I think.
Yeah, and you just touched on this as well, but I think it's so important for us as scientists to collaborate with other scientists in different fields because we're not ever looking at one thing in isolation, even in a greenhouse, right, where we can control almost everything. We're really not isolating living organisms down to one question that we can ask. I wonder if you can speak a little bit more about your collaborations in the research that you've done, either currently or in the future or in the past.
Yeah, I don't have a single project in my lab that is not a collaboration. Everything is a collaboration. And that phenotyping software that I started as a postdoc, started with another postdoc, and we still collaborate. So this is a long-term collaboration that's been going on since about 2013. So we're closing on 10 years now of this collaboration. And phenotyping in particular, we're taking images, and so it involves physics, it involves engineers. We're doing computer science, so obviously it's computational research, but there's biology involved, so there's biologists. If you look at the composition of just my lab, we have biochemists, we have molecular biologists, we have statisticians. We have somebody who, before coming to the lab, never touched a plant or worked on a plant before. He was purely a computer scientist working on machine learning. It's very interdisciplinary now, and it has to be because of the nature of the science that we're doing.
Absolutely. I couldn't agree more. And I too have no projects that don't involve other colleagues, for sure. There's a very specific question in the chat that I wanted to acknowledge and call out, and that is, are you considering using Vrix? It says spectrometers, but I think maybe, I wonder if that's spectrometers.
Yeah, I'm not familiar with that particular piece of equipment, but hyperspectral imaging is basically optical spectrophotometry. We have spatial information, so we know the wavelengths, and we also know each pixel, the profile of each of those pixels. So people can go in and spot check with a handheld spectrophotometer, but then we also, correlate that to what we see in the image data. I hope I answered that question.
Very cool. I'm wondering, it's kind of changing lines here a little bit. You talked earlier about preparing, the next generation of plant scientists, and I think that's another thing as us as a scientist really need to focus on that and make sure that we are, having people smarter than us and more productive than us following behind us, right, to continue the good work. And I wonder, what are some of the areas that your students are most excited about exploring?
Yeah, our lab in particular, we do a lot of education and outreach, and that's in part because we are very interdisciplinary, so everybody has to learn about what everybody else is doing in order to communicate. We've found that the phenotyping software that we have developed is a nice tool to teach people about data science and bioinformatics, which I think are very, very important regardless of what type of science you end up going into. So even if we have an undergraduate in the lab that may not stay in plant science, but may go on to do a different type of science, those data science skills, learning how to handle large sets of data are still going to be incredibly useful going forward, regardless of what lab they end up in or what field they end up in. So data science and bioinformatics is something we try and teach. And we have a collaboration at the Danforth Center with our education outreach, as well as Harris-Stowe University, to try and make a curriculum that is for data science and phenotyping in particular.
That's great. Yeah, I found that just teaching analytical thinking is so important for people in many different careers, if not all careers, really. So that's a really nice thing to do. Just thinking about, these people might not be scientists, they might not be plant scientists, but the things you're teaching them have so many applications, and that's really wonderful.
Yeah.
Yeah, go ahead. Sorry.
I was going to say, just learning to use evidence to reach conclusions, I think, is really important for people who are, voting. To say, look at this evidence and does it sort of match with what the results you're seeing are?
Absolutely. Another question from the audience here. Are you researching urban agriculture environments as well as rural farming? And I think maybe thinking about their differences in sort of urban ag plants, different plants might be more appropriate for urban settings than they are in rural settings.
Yeah, and so we are often trying to vary these environments, and again, we're working in very controlled settings, so we're working these boxes, and so we can set the environment to essentially what whatever we want to a point. So we can control temperature, humidity, light intensity. In some cases, the wavelengths of light that are reaching the plants. And so because we can do this, we can simulate various environments. We also collaborate with ecophysiologists who, are great at modeling. And so we can give them data in a particular environment, and they can then extrapolate the data that we give to them to other environments. So so that we don't have to test every single variation. And so modeling obviously is very important to the work that we do. And again, it's a collaboration.
Yeah, absolutely. And I think, looking at those all different conditions, and particularly in the work that you do in temperature, and thinking about urban areas are often heat islands, and so it might be really different environmentally, right? So that's really great. I'm wondering if you could talk a little bit about sort of how you've touched on this already a bit, but kind of how you communicate your work to the public beyond, of course, this program.
Sure. We do things like Twitter. So, if we have research coming out that we're excited about, we broadcast that through social media. I will plug, we have somebody in our lab, Katie Murphy, who has a really great TikTok where she actually shows, it's called Real Time Science, where she actually shows what it's like to work in the lab every day. So it's not sterile. So meaning that, sometimes it's challenging to do the work in the lab. It's not perfect every time. And so she shows the kind of day-to-day of working in the lab, which I think is great. We do workshops and we do things like this. We have an event called the Raspberry Pi Jam, where we bring kids in to sort of see the robotics and fun things that we have going on at the center, to try to bring a sort of younger generation in on the science that we're doing.
That's great. And I definitely want to come to the Raspberry Pi Jam. That sounds amazing.
We just had it a couple weeks ago, actually.
Oh, I'll come next year.
Yeah.
Fantastic. I see Emily has joined us again here, which means our time is up. This has been really wonderful. I want to just really thank you so much, Malia, for joining us today, and for the wonderful work that you're doing, and the great impact that you're having on our future ability to continue growing plants that'll feed the world. Thank you.
Thank you. And thank you for the invitation.